So our next part is the competitive selection of the oral presentations from the abstract submissions. So we have 3 speakers. The first is Dr. Rory Henderson. If you want to come up to the podium, and he will be speaking on the characterization of HIV Glycan V3 targeting DH270 broadly neutralizing antibody lineage.
That's fine. All right. Thank you, Georgia. So this work is work that I've been doing with Dr. Priyamvada Acharya, looking at one of our major vaccine targets at the DHVI, the DH270 lineage. And of course, on the trimer, there are several epitopes that broadly neutralizing antibodies target, among them the trimer apex, CD4 binding site, and the fusion peptide region. But our particular interest today is the Glycan V3, which is shown here on the trimer, adjacent to the apex. And this is the target for DH270. And so there are crystal structures available for the antibodies, and there are structures of the antibodies bound to a peptide mimetic of the V3 site. But the trimer structure is fairly complex, as you can see here. On the left, I have the trimer. GP120 is in blue, GP41 in orange, and then at the apex, V1, V2 in green, and V3 in red. And then I highlight the various sites, the apex CD4 binding site and the Glycan V3 site. But of course, on the right is the very dense glycan shield. And so in order for us to understand how this antibody has developed an ability to get through this glycan shield and to interact with the protein portions of the OMP trimer, it's important that we have the full context of the trimer in our structural analysis. The other thing that's complicating, a complicating factor with all of this is that proteins and the glycans are in constant motion. This is a molecular simulation at a relatively long time scale of the trimer. And you can see, especially if you look at this site, that the protein is differentially exposed. The glycans are actually moving in and out of a blocking configuration for the Glycan V3 site. So there are a lot of things to consider when we think about what we want to do to design a vaccine, especially in terms of those early encounters that the antibody makes with the protein. And so to this end, we asked the following questions. The primary question is, what are the key structural barriers to the development of neutralization breadth in the DH270 lineage? And to get at this, we ask, how does the HIV1 OM present the DH270 epitope when the antibody is not present? How does DH270 handle OM structural hindrances? And how does DH270 manage OM glycans? And all of this is made possible by a powerful microscope we have here at Duke now, the Titan Krios, which I show down here, that has made it possible for us to get many, many structures in a very short time. This project was started in March, and as you'll see, we already have a very large number of structures. But to get those structures, we take images. This is a representative micrograph of our protein, which are these small gray dots. And we take several thousands of these micrographs and crop out these particles, and we're able to generate these two-dimensional class averages that give us a little bit more high-resolution detail. You can see that we have different orientations of the trimer. And from this, we can build ab initio models and then refine those structures to high resolution. And the ultimate goal is to then fit atomic coordinates to those high-resolution details in the map that we can then use to identify different contacts and start thinking about how these antibodies have matured. And so the goal of the project is to get every structure in the lineage. This is the lineage here, starting with the unmutated common ancestor and going through several intermediates to reach the different mature forms. And in particular, we're interested for vaccine design in DH270.6 as it's the broadest and most potent antibody in the lineage. And this is what we have so far. We aim to have a total of 20 structures, so we have one more that we need to get. The resolutions are between 3.5 and 4.9. We aim to have these hopefully all below 4 and possibly around 3 angstrom, so ongoing work with that. I show up in this corner VRCL1 bound structures. So VRCL1 interacts with the CD4 binding site. And the reason that we're doing this is that since it's not in the DH270 site, we can see what that DH270 site looks like before the antibody is bound, because it's very relevant in a protein that's so flexible like the OMV to understand what does it look like before and what does the antibody have to force the OMV to change in terms of its shape. in order to bind. And so we have 5 structures from 5 different strains from the CH848 patient. These different numbers just identify different strains. As I said, of course, you can see that we have all the structures, but not only do we want to know what do the different strains look like, but also in the mature bound state, what do they look like in terms of the changes in the loops and glycans. And so we turn first to the question, what does the DH270 binding site look like before the antibody is bound? And again, this identifies the structural hurdles to antibody binding. This is for the CH848 1017 trimer, which is one of our primary immunogens right now. And in particular, we're looking at the DH270 binding site here, and V1 is a loop that we're very interested in. It's in this sort of lemon color. And what we found, the expectation was that it would be relatively disordered and it just sort of sits out and moves around. But we found that it was actually very well structured. You can see that there's clear density for this, and it's sitting over the GDIK motif, which is an important motif for a DH270 interaction. And so what this looks like is that the OMV is actually trying to protect that site from interaction with DH270. Looking at a different strain, we have the 0526.25. We find that the loop is very flexible. You see these missing densities. That's an indication that it's flexible. But what's really interesting here is not only is it protecting the GDIR motif in this case, but it's also reached over and is now interacting with the N332 glycan, which is critical for DH270 interaction. So this loop has actually changed conformation. It's a much longer loop than the previous one, and it's protecting a glycan in a way that we've never seen previously. And so just to bring that all together again, the CH848 HIV1 OM V1 loop protects the Glycan V3 epitope. The 1017 loop blocks the GDIK motif, and the 0526.25 loop protects both the GDIR motif and the N332 glycan. And we next asked, how do the DH270 antibodies adapt to the Glycan V3 binding site? The reason we want to know this is that changes between the unliganded and bound states explain how the mutations acquired by the antibody allow adaptation to the OMB surface. And so what I'm showing here is looking at the DH270 UCA in green. This is the heavy chain. And then again, you can see V1. What we find is that this conformation of V1, it's a different perspective than I showed previously, but it is the same conformation that it is before the antibody has bound. So in the case of the UCA, V1 has not moved, and it's preventing the DH270 UCA antibody from getting into close contact with the GDIK motif. When we look at the first intermediate after the UCA, we can see that the V1 loop has changed configuration dramatically, and we've identified this G57R mutation that occurred in I5 as causing this shift in the V1 loop. And so the antibody has learned to move this loop out of the way so that it can get into closer contact with the GDIK motif. So again, the DH270I5 acquires the ability to shift the CH848 HIV1 OMV V1 loop, and the DH270 UCA retains the unliganded V1 orientation with the GDIK motif protected. So we next asked, is the V1 shift in I5 accompanied by a shift in the orientation of the Fab relative to the OMV? This is important because a shift in the antibody binding site could have an important impact on the interface and therefore maturation toward broad neutralization. And the result here is that the V1 loop rearrangement in DH270I5 does result in a shift in the disposition. To identify this, we align GP120. There are actually 2 proteins here, although it looks like there's only one, because the GP120s align quite well. But if we look at the zoomed in picture, you can see the light color is I5, and these beta sheets are not aligning well with that. And if you look at the heavy chain, you can see that it's shifted more closely to the GDIK motif. So that shift of the V1 loop allows it to get in closer to the OMB surface, and it's able to pick up new and stronger contacts because of that. We next asked, how does the DH270 lineage manage the glycan shield? This is important because identification of key contacts with glycans provides clues to the mechanism by which DH270 antibodies improve their ability to penetrate the glycan shield. And so I show first the UCA to I5 transition. One of the important contacts that we see here is in the light chain. S27Y is interacting with N301, so we pick up this new interaction with the N301 glycan. Going from I5 to I3, we see another light chain mutation that is L48Y, and it actually picks up an interaction with N332. This is sort of on the opposite side of the antibody. And then going from I3 to I2, we can see out away from the CDRs, we have a mutation in the heavy chain, R85S, and that is changing the way the OMB is interacting with this N156 glycan. And so there are, of course, many other contacts and many other mutations that change the way that the protein interacts with the glycans, but we see that the N332 glycan is stabilized by contacts with the heavy and light chains, N301 glycan is stabilized by the light chain contacts, and the N156 glycan is stabilized by heavy chain contacts, distant from the antibody CDRs. And then final question that we wanted to ask is, how did the antibody conformation change during maturation? Antibody Fabs are known to occupy multiple conformational states that can impact binding. And so what we looked at in particular is a region of the antibody called the Fab elbow. I show that here. This is the rotational axis, and the elbow allows the FV and the constant regions to move relative to one another. And what we find is that in mature forms, going from 1 through 6, 1, 2, 4, and 6 have a similar elbow angle, but 5 and 3 differ fairly dramatically, especially between 6 and 3, which I show here. You can see it's a completely different orientation. So we do see that the differences are common in the lineage, and we know from a paper that we published this year that changes in the Fab elbow angles are known to impact the paratope shape and likely play a role in VNAb maturation. So in conclusion, structures for all DH270 antibody Fabs bound to the OMB trimer have been determined. The trimer V1 loop protects the DH270 binding site. The early intermediate antibodies learn to shift the V1 loop. Key glycan contacts stabilize the DH270 bound state. And the DH270 antibody conformations and dispositions relative to the OMB trimer change as the antibody matures toward neutralization breadth. And so with that, I'd like to thank everybody who helped with this project, especially Priyambada Acharya and members of her lab. And I'd like to thank our funding. Thank you.
Thank you very much, Rory. Can you comment on, you mentioned that the orientation of the V1 loop in 848 is rather unusual. Do you think applying this to vaccine design going forward, how common will it be that you need to design an immunogen that can elicit an antibody that can move the V1 loop out of the way of its binding site?
So we've started to look at all of the Glycan V3 sites, or Glycan V3 VNAbs, and we are seeing some shifts in the V1 loop. I think this one in particular is different than the others because of how strong that protection is. Usually if we look at the maps, we see that the V1 loop is relatively flexible. And we have some additional V1 loops that are more flexible in the CH848 virus that are more flexible. So it's really these two particular ones that I showed that seem to have this strong protection that DH270 had to learn. In principle, of course, the virus could learn to do that for any Glycan V3 targeting site, but we haven't seen it.
Great, thank you. Any other questions for Rory? Celia?
So, were your structures that you made looking at the unliganded binding site with DH270, were those structures with PRC01 ligand envelope? And if so, would it be important to look at an envelope ligand by a different antibody to see if there are structural constraints that are conferred by PRC01 ligand into the envelope that might make the conclusions a little bit different or with another structure?
Yeah, so everything we have right now is for VRC01 for this particular project. It's definitely important to consider that VRC01 could have changed something, but we do have some other data sets. Because of the way cryo-EM works, if we have heterogeneous population of bound and unbound trimers, we can actually separate those out. And looking at those other structures, we see trimers that have nothing bound to them. And in the case of 1017 at least, the conformation of the V1 loop is the same. So there's some evidence there, but definitely it will be important as a control to show that using some other antibody as a fiducial marker that we can that we capture the same state, that it's not induced by VRC01.
Thank you so much.
Thank you.
Thank you again, Rory.